The exact causes of Alzheimer's disease are unknown. However, it is known that amyloid beta peptide toxicity is involved in Alzheimer's, but the mechanism of its contribution has not been worked out. The authors wanted to work in yeast because of the unequal toolkit and the attractive discovery platform offered by the yeast system. Amyloid beta, A-beta, is formed through processing of a precursor in the secretory pathway, and the most toxic form is A-beta 142. A-beta oligomers have been shown to increase endocytosis in cultured cells, and fibroblasts from AD patients have been shown to exhibit defects in endocytosis. Yeast are fundamentally different from neurons, but they offer experimental advantages, such as powerful genetic techniques and rapid growth. Therefore, the authors first wanted to determine if A-beta toxicity could be modeled in yeast. To do this, they used the most toxic form of A-beta, 142, and observed cell growth. They found that A-beta contributed to toxicity in yeast, as in neurons. Next, the authors wanted to identify genes that modulated A-beta toxicity. They transformed a screening screen with 90% of the full-length open reading frames found in yeast driven by the inducible GAL1 promoter. They induced in different medias to identify enhancers and suppressors of A-beta toxicity. They identified 23 suppressors, 12 had human homologs, and 3 had known functions in endocytosis. They chose to continue with the 3 that had known functions in endocytosis and had been implicated in risk for Alzheimer's disease. One of these is PICOM. PICOM, phosphatidylinosyl-binding clathrin assembly protein, interacts with clathrin and has been highly confirmed as a risk factor for AD in humans. The authors wanted to determine if PICOM was able to protect neurons from the toxic effects of A-beta. They used cultured rat cortical neurons. It should be noted that we took artistic license and A-beta is not secreted, although it may appear to be in our demonstration. They found that infection with a lentiviral PICOM construct was able to partially protect the cells from the toxic effects of A-beta. The next question they wanted to answer was if A-beta affects clathrin distribution in yeast and therefore alters endocytosis. They tagged endogenous clathrin light chain with YFP and observed localization. In control cells, they observed the expected distribution of CLC1 foci. In cells expressing A-beta, they observed perturbed clathrin localization with increasing number and brightness of foci but a decrease in their average size. They suggest that this indicates a defect in clathrin-mediated secretion and endocytosis. To specifically look at the effects of A-beta on CLATHRIM-mediated endocytosis, they tagged STE3, which is a G-protein-coupled receptor that is constitutively endocytosed and degraded in the vacuole, with YFP. In control cells, they observed normal localization. In A-beta expressing screens, they saw a reduction of delivery of STE3 to the vacuole. From this, they concluded that endocytic trafficking from the plasma membrane was profoundly disrupted. PICOM was able to almost completely reverse the defective endocytic trafficking caused by A-beta. They also tested the 2 other genes found to be A-beta suppressors with functions in endocytosis and saw a partial reversal of the disrupted trafficking caused by A-beta. The authors were able to model A-beta toxicity in the yeast system. They were able to confirm the relationship of A-beta in yeast and rats. They showed that endocytic trafficking is disrupted in response to A-beta toxicity and identified conserved suppressor genes that alleviated the trafficking defect. Neurons are particularly vulnerable to disruptions in the homeostasis of endocytosis because they are constantly recycling neurotransmitters and their receptors. Few treatments exist for AD and the problem of rescuing neuronal functions has yet to be solved. Their yeast model provides a tool for identifying genetic leads, mechanisms of action, and screens for potential modifiers of this complex disease.